hari ini. Oke, Oke. Terima kasih kepada seluruh peserta yang sudah menyempatkan waktunya ya untuk bisa mengikuti kuliah tamu pada hari ini. Sebelumnya mungkin bisa nanti apa dibuka link yang dari admin ya untuk mengganti background untuk mengganti background eh, Zoom Anda ya. Jadi mohon nanti diganti sesuai dengan acara kita pada hari ini, gitu ya. Ada 20-an mungkin ya, Destri ya, yang hadir saat ini. Sebelumnya saya ucapkan uh, selamat pagi kepada Bapak dosen kita ya, Pak Indra ya. Selamat pagi Pak Indra. Uh, Terima kasih sudah hadir pada kuliah tamu hari ini dan juga Bu Destri. Terima kasih ya sudah apa ya mengarrange kuliah tamu hari ini. Mungkin kalau nggak Bu Destri, mungkin kita nggak akan bisa kedatangan tamu dari Singapura gitu ya. Oke. Eh, Alhamdulillah ya, ini sudah mulai banyak yang datang. Mungkin bisa saya mulai saja. Nanti eh, selama kurang lebih 1 hingga 1,5 jam ya, saya Andita Imandasari sebagai moderator pada kuliah tamu hari ini akan memandu jalannya kuliah tamu pada hari ini selama kurang lebih 1 hingga 1,5 jam, gitu ya. Nanti teman-teman di sini para audiens nanti bisa bertanya setelah eh, pembicara menyelesaikan materinya, begitu ya kira-kira. Oke, kemudian sebelum kuliah dimulai ya, mari kita berkenalan dulu. eh, Guest lecturer kita pada hari ini yang luar biasa, yang sudah didatangkan jauh-jauh dari Singapura, tepatnya nasional, nasional, National Healthcare Group Diagnostic Singapura, gitu ya. Good morning for Mr. Hong Fat Lee.
Good morning.
Okay, thank you. Welcome to our Faculty of Health Science, Universitas Maarif Latif Sidoarjo, Indonesia. And my name is Adita Imandasari. Uh, as I will guide lecture today. Before the presentation begin, I want me to introduce our guest lecturer to you. Mr. Hong Fadli is Senior Medical Laboratory Scientist from National Healthcare Group Diagnostic Singapore. He had been worked in hospital in Malaysia and several hospitals in Singapore before he works in NHGD Singapore. Yeah. He has been amazingly working in a medical laboratory field for 17 years. So we are undoubtedly his experiences in the medical laboratory field. And the audiences are so lucky can be here to learn the development of techniques in hematology, especially from a molecular perspective. Jadi, pembicara kita kali ini didatangkan dari Singapura ya, para peserta. Nah, semoga nanti teman-teman dan peserta semuanya di sini bisa mengikuti kamu dengan baik. Nanti jika ada pertanyaan bisa disampaikan setelah presentasi materi oleh pembicara kita selesai ya. Jadi ada uh, uh, 1 jam ya hingga 1,5 jam dengan sesi tanya jawab untuk 3 pertanyaan. So, Mr. Lee, this guest lecture today will run for 1 to maximum 1.5 hour with along with Q&A session for 3 questions from audience. So you will deliver your topic to audience and we will continue for Q&A sessions after you finish your topic presentations. Well, Mr. Lee, time is yours.
Thank you. Okay, terima kasih Miss Andita. Okay, for the introduction. Okay, and also terima kasih kepada University Maarif Hasyim Latif for this invitation for the talk. Okay, selamat pagi semua. Okay, and terima kasih kerana spend your precious Sunday morning with me. Okay. So today I'm going to give a talk about the molecular techniques. Okay. In the role of molecular techniques in hematology. Okay. Okay. I will share my slide first. Hmm. Okay. I have quite a lot of slides to share, so we will start now. Huh? Okay, today the learning objective is will give some introduction to recent molecular techniques. Okay, and then the learners will be able to know the importance of the molecular techniques in hematology and how it applied in this hematology related. Okay. So the roles of the molecular techniques nowadays it provides the advanced information, okay, of diseases related to the blood facilitating diagnosis. So especially in a treatment choice, the prognosis means a survival rate and the response follow up. So this the example of hematological related field is like malignancy. Okay, the most common one is leukemia, lymphoma, and myeloma. Even though thalassemias, hemoglobinopathies, hemostasis, and even though thrombosis. Okay, let's see some statistics. Okay. So in 2022 statistic in US, you see from this statistic we can tell that actually cancer is the second main reason of the death in US. Okay, just after the heart diseases. Okay, we further break down of the cancer statistic. Top 10, we will find that always on the board is a non-Hodgkin lymphoma and leukemia. Okay, which is a hematological malignancy related. Okay, the other is non-related. To this, today we will focus more to this hematology area. So we find in male around like 80,000 new cases, okay, in 2022, and around 60,000 of this malignancy in US, okay, for female. Even though death also quite significant, okay, it's talk about like 25,000 death, okay, by this leukemia and non-Hodgkin lymphoma. For female, around also 20,000. So from the statistic, actually, as a guy, you are more unlucky. So you have higher rates. So this is the one good thing to be a female, right? So we come back to Indonesia. Okay, I can find the statistic in 2018 only. Okay, actually the trend is almost the same. Okay, we also find that leukemia and non-Hodgkin lymphoma in the top 10 of hematological, that is cancer cases. Okay. So actually, this is it's a worldwide trend. Everyone better remember to unmute if you are not going to raise your question or any concern. Thank you. So we further go to see the national trends in a cancer death rate. Okay, actually the good thing is a declining rate, so it's decreasing trend. You see, even though myeloma, this lymphoma and leukemia is all at the decreasing trend. So it's a sign of during the advancement of these molecular techniques. Okay, a lot of research have been done, right? And then it improved the help in diagnosis, and even though the survival rates and treatments for this kind of patient. this statistics is pulled out from the Cancer Society from US. Okay, since the top 10 usually cancer for hematological related is a cancer. I will give some example of the oh, so sorry. I'm so sorry. Just now I didn't unmute, right? Okay. I will start from beginning again. So sorry for that. Yeah.
Oh, it's okay. I think you can like maybe from the statistic.
Okay.
Yeah.
Hmm. Hi. So sorry. Today, my topic is the role of molecular techniques in hematology. Okay. Learning objective is we're going to have an introduction to some recent molecular techniques. The learner will know the importance and the application of these molecular techniques. So sorry that just now I forgot to unmute. Thanks for reminding. So the roles will be the provide advanced information of the diseases related to the blood facilitating diagnosis, treatment, the survival rate, and response follow-up. It involves a hematological malignancy, thalassemias, hemoglobinopathies, hemostasis, and thrombosis as well. Okay, so this let's see some statistics. Okay, we will find that in 2022 in US, cancer is the second main causes of the death just after the heart diseases. So we break down further in this cancer type, okay, top 10, we find that for hematological related is non-Hodgkin lymphoma and also leukemia, always on the board. Okay, so around 70,000 of 70,000 of male for the new cases, and a female is around 60,000. And even though death cases involve 25,000 of death for male and around 20,000 of female. So from this statistic, actually, as a guy, you are very unlucky. So you have a higher risk, okay, for this type of malignancy compared to a female. So this is another good thing to be a female, right? So we come back to Indonesia. I found a statistic in 2018. So leukemia and non-Hodgkin lymphoma is still the top 10 from all types of cancer. So it's a worldwide trend. Okay. Now we take a look on the this trend the death causes by this cancer trend. Actually, this is a good thing that is on the declining rate. So it means decreasing. Okay. So thanks to the technology in my molecular techniques, is not the maybe not only the factor, but it's one of the factor when the advancement of these molecular trends, they did a lot of especially the recent development, a lot of more researchers. It helped in diagnosis, even though the method. So and also help the to improve the survival rates. Okay, and how they follow up for the patient. Okay. And that's why it's a dropping trend. This significance of this hematological related cancer in this causing the death. Okay, so I will give some example of these cancers. like ALL, acute lymphoblastic leukemia, acute myeloid leukemia. CLL is a chronic type, CML, and then others type of MPD, such as primary myelofibrosis, essential thrombocythemia, and even though polycythemia. Other lymphoproliferative syndrome, such as Hodgkin and non-Hodgkin lymphoma, and then multiple myeloma, and even though MDS. Okay, come to this photo. Okay, it's after CBC. Okay, a staff go to do a blood film staining lah. This is a basic hematology test. Okay, we found that a patient having the marked leukocytosis, anemia, and also borderline low platelet count, and then have abnormal deep flagging. So anyone want to try to identify this cell? You can type or unmute yourself so you can answer. I will wait for 10 seconds. See anybody want to try?
Mungkin teman-teman di sini ada yang mau menjawab, ya.
Yeah.
Kira-kira
So
ini
anybody?
diagnosisnya apa? Ada nggak? Mungkin maunya dikasih pulsa.
Unmute yourself. Maybe can. Yeah. You can answer in a chat. In the chat if you are shy to talk.
Ya.
Yeah.
Ya. Silakan audiens, para peserta kuliah tamu hari ini boleh menebak ya. Boleh menebak tadi itu diagnosisnya apa di chat.
Anyone want to try to identify?
Ya. Mungkin malu untuk ngomong bahasa Inggris, jadi kalian bisa tulis saja di chat.
Nah, tulis juga boleh ya.
Tulis di chat, silakan. Ada yang mau mencoba untuk menjawab kira-kira ini diagnosisnya apa jika gambarannya seperti ini. Mungkin dari teman-teman intensif atau ahli jenjang ya, yang mungkin sudah berpengalaman kerja di lab diagnostik, boleh. Ada hadiahnya nggak ini Bu Destri yang menjawab? Nggak ada.
Ada poin nilai ditambah.
Poin nilai. Okay. So if you can, if you want to try to identify this diagnosis, this
Okay.
yeah.
I think never mind because I think everyone is shy. Okay. So this cell is a blood cell so by morphology wise is a nuclei. Okay, high NC ratio. Okay, large size of the cell is an abnormal cell. We call it as a blood cell. It's a sign of leukemia. Okay, so by this only this information, is it enough for you to make the diagnosis? Even though like a specific diagnosis cannot, right? Because there's actually quite many types of malignancy, and also you cannot just specific the treatment plan for this patient as well. So that's why we need other more specific tests, such as molecular techniques. So molecular diagnostic tests can be a genomic, a DNA, or RNA based. Okay, so genomic, we usually talk about cytogenetics and karyotyping and FISH. Okay, DNA based is a PCR, very common test, polymerase chain reaction. RNA is the reverse transcriptase PCR, and then Sanger DNA sequencing as well. Okay, or nowadays we are using the modified Sanger DNA sequencing. Okay, and then the new generation sequencing as well. Okay, later we will go through one by one. Okay, in brief because we will concentrate more to the application. Okay, sample types is the test dependent. Okay, usually for hematological related problem will require peripheral blood. Okay, If for malignancy related, is preferable more than 5% of blood. Okay, and also bone marrow is preferred for this cancer detection. So some basic of a DNA mutation. It can be a point mutation, chromosomal mutation, and copy number variation. So point mutation can be a substitution, insertion, or deletion. It's very common. You can find in even the book. Chromosomal mutation, it can be an inversion, deletion, duplication, and translocation. So copy number variation can be a gene amplification, expanding to trinucleotide repeat. Okay, we give a point mutation example. So we talk about like a single base mutation. Okay, in the cells, sickle cell anemia, involved in this HBB gene. So you see, if there is the A sequence, the base replaced by this T, it become coded for the wrong amino acid. So if this amino acid shifted, okay, change will cause the morphology change in the of the membrane as well, because why it is a protein, right? If a change in the conformation, it also will alter the membrane of the RBC lah. Okay, so the picture also show that this is a sickle cell anemia. Okay, the RBC is very abnormal. Okay, now come to this chromosomal mutation. Okay, we use a very famous model, Philadelphia chromosome. It involves the translocation mutation of the chromosome 9 and chromosome 22, which is almost can find in this chronic myeloid leukemia patient, and also the quite common in Philadelphia positive ALL, acute lymphoblastic leukemia patient. For CML case, the major isoform will be the P210, okay, as a fusion protein, and the minor minority will be the P190, or we call it as P185. This fusion protein, okay, the isoform is a product of when the ABL gene, the breakpoint, is different from the cutting site. So you see where we see the ABL gene from a chromosome 9, it has been translocated to this BCR gene, and it combined. Then it coded for a mutated fusion protein. So the difference of this Philadelphia positive ALL, actually the fusion protein is reversed. Is the major form actually is the P190, and the minor form is P210. This information, this isoform, if you can know it, actually it helps in a doctor to predict the treatment efficiency, effectiveness later. Because any research paper proved that P210 actually is a hallmark of CML, and P190 usually have a more resistance to the drugs. So we come to this molecular techniques. Okay, maybe some of you already know. So I will just go through a few briefly. If any question can raise later. So basically, we start with the nucleic acid extraction now. Okay, usually there will be a one-step lysis method and two-step lysis method. Okay, two-step lysis method is actually like okay, for hematology related, usually required blood sample. So that's why I'm focusing on this one. So usually the first step is lysing the RBC, second step is lysing the WBC and all other items. Okay, after the digesting treatment, so you remove the protein. Okay, then you recover the DNA basically. And then the one step will be the everything lysis will be happen in one step. Then continue with you contaminant removal, that's almost the same. Then also recover your DNA. So this classic type, some lab very seldom using this manual method already because especially in hospital lab, the sample is continuous coming. So most of the labs will not use this manual method. Usually they now have automated. Okay, I just introduced to you that actually in the market a lot of automated nucleic acid extraction. One of the leading company is like Chargent. Okay, even though they are automated instrument able to extract the DNA and RNA as well. Okay, according to the user requirement. Okay. So we talk about next molecular techniques. Okay, it's a karyotyping. Okay, I'm sure you hear about it. Okay, it's very common. So actually, this is a technique that can analyze for chromosome abnormalities for the cause of malformation or diseases. Okay, so basically we count the number of chromosomes and look for the structural change in chromosome as well. Okay, to detect the abnormality. So human usually have 23 set of the genome chromosome. So you see from 1 to 22 is the autosome and one sex chromosome based on you are female or male, X or Y. And then after we stain with this, usually we use the ginseng to stain. And this photo is showing after you stain with ginseng stain, you detect by the bright field microscopy and use the software to detect the abnormality. Actually, the software quite user friendly. It will automatically analyze for you. So this karyotyping steps involve sampling, then culture for around 3 days. Then you add this culturing and hypotonic saline. So the cell will maintain at the metaphase. This metaphase usually the chromosome will be more condensed and also aligned at the center of. And then after this, we need to fix at the slide and digest with trypsin to free out these chromosomes and stain with ginseng, then analyze it with the software. I will give an example. So for karyotyping of Philadelphia just now, what I told is a very famous model especially for education. Okay, you see the chromosome 9. Actually, two chromosomes are not identical. One is longer. Okay, another chromosome 22. Another set of chromosome is shortened. So this is a sign of a Philadelphia problem. Okay. So in Singapore, for this karyotyping, actually is also being offered. Okay, especially for this cancer chromosomal karyotyping. Okay, also required mostly bone marrow specimen type is peripheral blood and even though lymph node. Okay, so just now we talked about the karyotyping, right? It involves the culture. So you spend actually wasting a lot of time for the culture to be done. Usually it will be done in 3 days. Okay? But this fish method, okay, this fish is not the one you cook okay, in your home. It's a frozen in situ hybridization. So it's used the frozenly labeled molecular groups to locate the specific DNA sequence. Okay? For diagnosis of the genetic diseases, especially cancer. So the timeline, when you look at timeline, actually the first DNA, RNA fish experiment tested by this Gillespie and Spiegelman. Then after this, the first in situ hybridization using the RNA probes by this Gal and Badu. Okay, then the advancement keep continuing on, especially to the probes design technology improve. Now we have so many types of probes and even you can design your probes in a even sometimes different multiple colors, okay, according to your needs. So fish advantages is can use the metaphase or interphase chromosome because no cells culture are required, okay, because it's specifically detected the DNA sequence that you are interested. So we use the labeling probes with different profiles allow the detection of multiple sequence at a time. So it also can detect multiple target sequence if as long as you provide different probes, okay, for this process. So fish step will involve you just provide the DNA probe, okay, with a label with fluorescently and target your target DNA. After you denature your DNA from double-stranded become single-stranded and let it hybridize each again. So your DNA probe will be able to find to the target sequence. Then you can visualize by fluorescent microscopy. So this diagram are showing is to simplify the process. So you see the probe DNA labeled by the fluorescent, then it will bind to the target sequence and make the gene in the chromosome, the interested part will be fluorescently detected. Okay, examples again. So we use FIKES Philadelphia translocation mutation to compare. So we can compare to the previous method. Okay. So as a normal cell, from the left side photo is a normal cell with a pure green signal, only green signal and red signal. So the green signal is representing the PCR, okay, and then the red signal is a chromosome 9 ABL, okay, probes label. So if there's no mutation, only red signal and green signal will be available. So we observe the abnormal cells at the right photo. You will see a fusion signal which is you see green, you will see red. So in one area, so it's a fusion signal means the PCR and APL mutation happen This fish groups panel actually also been offered in Singapore. For the common types, usually will be a multiple myeloma detection. There's a lot of mutation sites. It will be detected together in one run. So for myelodysplastic syndrome as well, and also the CLL panel, you see, you can use multiple probes to detect the multiple mutation sites. This table is just give some extra example for cytogenetic abnormalities for T cell ALL, okay, acute lymphoblastic leukemia. A lot of mutation sites can be done by this cytogenetic testing. And this table is talk about the B cell ALL. I will not go through the mutation site one by one because it's very dry. Okay, so I just to highlight you actually the advancement of this molecular techniques. You see, it helps more mutation sites to be found out. Okay, and I'm sure this table will be growing in the future. Okay, next one will be the PCR. It's very common method. Okay, this method is accurately, efficiently, and rapidly amplify the small amount of DNA sample. So even though you have one DNA sample, actually you can amplify to become can be more than 1 billion copies of DNA that you are interested. Okay. So this diagram is just simplify the process which I'm going through in this PCR. So from denaturing from double-stranded DNA to method become the two single-stranded DNA, then annealing with the primer, and after this is the synthesizing steps by the stacking of this DNA polymerase. Then it initiates the copy of the single-stranded DNA to get your multiple interested DNA sequence. Okay, friends. And this cycle will be repeated in 20 to 40 times. Okay. Okay. This PCR timeline again. So actually, the first polymerase chain reaction was invented by Maarif in 1984. Okay. After this, the real-time PCR, okay, reverse transcriptase PCR is enabled, okay, in by this Higuchi 1993, and after this, it also pushed the for the what we call the NGS next generation sequencing to be in development until now is quite common as well. Okay, so PCR advantages and disadvantages. Okay, for the advantages, baik lah, the baik adalah these are highly specific. Okay, because as long as you provide the specific primer, it just target the segment that you want. Okay, and it's sensitive. From one copy, you can get more to 1 billion copies of DNA. Versatile, is adaptable, and rapid and efficient. So the disadvantages is it have a chance to be contamination. Okay, and then the cost and the complexity, especially the cost, because nowadays if you talk about the molecular methods, the charges will still be more expensive, and even the reagent, the kits is very expensive compared to the other area of the test, like basic hemato, basic chemistry, or even though microbiology test is more costly. By PCR alone also lack of novel information. Okay, because by PCR you don't know the sequence. Okay, you cannot tell much more info. And inhibition from the sample content. If you didn't purify your nucleic acid well, actually it may inhibit your affect your endpoint quality. Okay, so there's a lot of PCR types in the market. Now we can compare like what is PCR with real-time PCR. Okay, and also we can talk about the comparison of the PCR with reverse transcription PCR. Okay, and also this quantitative PCR with the digital PCR, which is more widely used compared to previous time. Okay. So for this reverse transcriptase PCR, actually is talking about reverse transcription. It usually convert your mRNA to become your complement DNA and analyze it in the DNA level. Okay, so it shows the promise as a means of detecting low levels of cancer cells. A lot of research papers have been proved that. Okay, for this reverse transcription PCR, it involves always two steps. Step one, it will be the use of the reverse transcriptase, okay, to reverse transcribe from the mRNA to become the cDNA. Okay, once you get your cDNA complement DNA, then you will just carry on the PCR as usual. Okay, to get multiple sequence of your interested DNA fragment. Okay, so this diagram just to show you the basic of different of DNA versus RNA lah. So from RNA actually the base different from the thymine and change replaced by this uracil. Okay, and DNA is a double-stranded. Okay, nucleic acid. RNA is just a single-stranded. Okay, nucleic acid. Okay. So the reverse transcription PCR is also widely used in a hospital setting. One of the example I give is like qualitative PCR, mRNA detection. No? You see the TAT also will involve 2 to 5 working days for this isoform P210, just I mentioned, and extra long 14 working days, almost 2 weeks to identify this P190. So now we compare the endpoint PCR versus qPCR. Okay, endpoint PCR means you cannot quantify your copy numbers during the PCR process. And the qPCR is a quantitative method, and nowadays we will always call it as a real time. So many people always confuse RT-PCR. RT-PCR, sometimes people may think that is a reverse transcriptase PCR only, but actually it's a real-time PCR. So nowadays we always describe this method as a qPCR. So it's a quantitative real-time to differentiate with the reverse transcriptase PCR. So definitely reverse transcriptase PCR also can be a quantitative method. Okay, so we also can be called as a RTQ PCR. So the endpoint PCR, you will need the gel electrophoresis at the completion of your PCR. Okay, then you have to use the densitometry methods to quantify your copy numbers. But this qPCR you definitely no need because using the method of fluorescence detection. So there's a free flow fluorescence the things dye lah, we call it dye at the mixture. Okay, once the DNA synthesized, it will capture the dye, okay, fluorescent dye, and when it accumulates more, it direct proportional to the copy numbers, okay, to your quantity. So usually it will request to okay, later I will give more example. It will require a standard curve, okay, to quantify your copy numbers. So definitely endpoint in the end, you will need longer turnaround time, and qPCR will shorten your TAT. So we talk about digital PCR as well. Okay, this digital PCR actually since 1992 recognized by this SKYS as a limiting dilution PCR. Okay. So usually what we do is the method using you diluted the sample and will random distributed to multiple partitions. Okay, can refer to this diagram. Okay, after we prepare the sample dilution and the PCR reaction mixture. Okay, so it randomly distributed to these multiple partitions. Okay, so some of the partition may have your interested DNA, okay, and some of it is empty, and some of it is maybe have more than one DNA that you interested. Okay, after this, we just doing the PCR as usual at different partition. Okay, so the PCR is happen in every single present DNA present partitions. Okay, and it will be detect fluorescently for each partition. You see, this method is good in you no need to base on the standard curve in a quantitative PCR. From the start of the PCR, as long as the fluorescence detection can be done, it already can provide you the copy numbers. Okay. So the methods can be a nanoplates, droplets, microfluidic plates, and microfluidic chips. This method also will apply a Poisson laws, okay, to help in the accuracy of quantification. Okay, because this Poisson law is talked about. Okay, let's say you have a lot of basket in front of you and you blindfold yourself, you throw the ball. Okay, the ball will be randomly go into one of the basket. Okay, if you continue to throw, it will maybe some of the basket have one ball and some may not absence of ball, and some maybe have more than one. Okay, so this random distribution, the mathematician put into the consideration. Okay, so what they do is they're using a simple mathematics. So valid partition is example, they use the total partition that you have. Okay, like let's say you have 8,000 partition. Then the positive partition means the one only with the presence of DNA. Okay, so you minus and divide by this total partition again, you will get your calculated target copies. And from this result, you just divide by the volume per partition. Okay, this is very specific lah. You can refer back to your instrument information, because different instrument can have different volume per partition. Then you can back calculate and get back your copy numbers per microliter. So this one you see it look like a bit, maybe some of you may think that this is very complicated, but actually it will be auto calculated by the instrument as well. So qPCR versus this dPCR, digital PCR. You see this diagram showing, so using the bulk reaction analysis for this real-time PCR. So you need to construct a, we call it standard curve. Okay, using the known concentration of the your standard DNA as a reference. So then you compare that, only you can know they give you the quantified result. Okay. But digital PCR, it just detects the absolute quantification from the detection of this fluorescence, whether it happened in which partition, the total partition that is detected. So it's faster in giving you the result, and also more precise. So if the real-time PCR can detect as little as the two-fold changes, but this digital PCR definitely will be a lower limit of detection. And usually digital PCR is preferred by the user, especially to quantify the real targets. The problem in this real-time PCR, actually they use the standard. Maybe the length of the DNA is a bit different with your target DNA. So it may have some effect, okay, in the precision. Okay, so if the rare target, which is rarely, especially for research purpose, your target gene is a new seldom have a research paper to target, and quite new in the market. Okay, so they would prefer using this digital PCR. So this digital PCR also can be used in a Singapore hospital lab, especially for JAK2 mutation. Okay. Okay. The application of this JAK2 mutation, especially in MPS, okay, myeloproliferative disorder. One of the example is PV, polycythemia. So usually the patient will have a very high RBC, PCV, and hemoglobin level. So usually also very also leukocytosis, thrombocytosis. Okay, then 95 to 98% of these patients will have this JAK2 mutation. Okay. So another types of MDS, essential thrombocythemia, we call ET. Okay. This is not the alien ET. So it's a type of disease. Okay. So usually it's the excess number of the platelet lead to the abnormal blood clotting and block the flow of the blood in the blood vessel because of the too many platelets in the bloodstream. So this JAK2 mutation will happen in 50 to 60% of this patient, ET patient, okay? And to differentiate further from the just now the polycythemia patient, you can also detect the CALR and the MPL gene, which is CALR is more common with 46.9%. So with these two present mutation, you can sure that it's not just now the polycythemia. It's a higher chance it's an essential thrombocythemia. So look at the bone marrow. I just show you for extra information for bone marrow of this ET patient. Usually you will see a lot of megakaryocytes. Okay. So another type of application is primary myelofibrosis. Okay. So also have the JAK2 mutation again, same 50 to 60%. Also have CALR, but less common, and MPL gene. So don't worry, we will still not mix up with the just now the ET, okay, the essential thrombocythemia, because you can use it together with the other basic hematology tests like a pen. You can know that patient is a pancytopenia, low WBC, low HP, and low platelet count. Okay, and the bone marrow usually is showing dry tap. We call it. Even though you doing the aspiration, bone marrow aspiration, always doctor will say dry tap. You cannot get a good example sampling for the bone marrow. Okay, next we will come to this DNA sequencing. Okay. So using the PCR itself, we cannot know the sequence of the nucleotides, but by sequencing, yes. So you can know the sequence of A, T, C, G bases in the DNA sequence for further analysis. So let's see this timeline again. Okay, from 1977, the classic type of Sanger sequencing method is invented. Then it triggered the starting of a genome project initiated in 1990 to 2003. Once this human genome project carry on, a lot of research has been done about this molecular finding. It helps in diagnosis again, even though treatment, targeted therapy, and improving the survival rate of the patient very well. So now, even though okay, it's okay. So we will see more the how it look like. Nowadays, we don't use any more classic Sanger sequencing. Most of it usually modified Sanger sequencing. So they use the reaction mixture with the primer, the DNA, and then DNA polymerase, same like a PCR, Then they provide the DNTP, what we call dideoxy nucleotide, okay, which the common one, the normal one is there's a hydroxy group, okay, at the 2' position. And then also another DDNTP with a fluorochrome's label. You observe this DDNTP, there is no hydroxy group at the 2' end. Okay, so when the new nucleotide, this DDNTP, go to bind to the DNA strand, the new strand, which is newly synthesized, so it will be terminated because there is no binding site for this OH, hydroxy. So it will be terminated. So from this result, you will create a lot of different fragments with different size of DNA created. So after running through the gel electrophoresis and detected by this laser for this because of fluorescent label, it will give the specific peak of color. Okay, same like this peak is represent a T, then it's T, and then C, T, A, like that. Follow the peak trigger. Okay, one of the application for this DNA sequencing is one of very important IBL related is a kinase domain mutation screen, especially important for the patient with drug resistance, okay, which we will touch later. Okay, so, okay, now come to this new generation sequencing. Okay, also talk about sequencing of DNA, but it talk about massively parallel DNA sequencing, ultra high throughput, okay, with a scale ability and a speed. It means it even can sequence for entire genomes. So for DNA and RNA, it involves always in the three main things. The chemistry involved, such as sequencing by ligation, synthesize, nanopore. For detection, is optical, solid state detection, even though electrical detection. Yeah, sorry. For amplification wise, is for second generation sequencer is by emotion PCR or by bridge amplification. Okay. For the third and fourth generation sequencer is more famous one nowadays is SMRT and nanopore. Okay. Okay. For second generation new generation sequencing is showing it in this diagram. So chiral sequencing and this ligation method actually due to the limitation, a lot of limitation and disadvantages compared to other methods, especially this synthesized method. Usually, especially this Illumina brand sequencer using this bridge amplification is more widely used now. So what they do is, so they from your whole DNA sample, you have to fragment it down using a restriction enzyme. Okay, so then you adapt end repair to end repair, and with the adapter ligation, so the adapter is helped for it to bind to the flow cell of the bridge and PCR amplification later. Okay, to hold your interested DNA so it won't be washed away, and PCR as usual, then for further sequencing. Okay, another second gen NGS method is ion torrent. Okay, this method is using the sequential of flood of DNTP. Okay, so it detects the release of hydrogen ions. Okay, the thing is when the new DNTP added to the new strand of DNA, it will release the pyro phosphate and also estrogen, hydrogen ions, okay, which will make the pH a bit changes, okay, pH change, and is detected by this ion sensor. So you see example, the green one if represent is A, okay, let's say it's A. So when it's turned for the DNTP A bases flood to the this sample, so it will if is the sequence can be added, it will trigger, it will release hydrogen ion, and it will create a peak detected by this ion sensor. So if let's say later the next sequence is T, okay, for this T, if there's a two T sequence added, okay, at the same time, it will trigger double pH effect and detected by this ion sensor. So you know, oh, after the A is 2T. Okay, let's say the next one is a red. Red represent G. There's no reaction. So you know the next sequence is not G. Okay, so continuous like this. Okay, there's a A, T, T, no G, and then the next one is still A like this. The sequencing will be carry on like this. Okay. So this second generation sequencer examples in the market, I will just for your reference. So for Illumina brand and also the Ion Torrent, which is more popular in a hospital setting now. Now talk about some more future related sequencing, third generation sequencing. Okay, this especially this MRT method by Pacific Bioscience. So it uses a concept of a zero mode waveguide, which is a lot of tiny wells, which can only carry one DNA molecules, okay, immobilized at the bottom of every single wells. Then it will be very sensitively detecting the fluorescently labeled nucleotide. Okay, so what they do is they use a adapter which is a hairpin type for your double-stranded DNA. Okay, so it will after you melt it to the single-stranded DNA, it will become a single circulated DNA. When during the PCR and sequencing, it will make it a repeated uh, repeated sequencing, and keep compare each other. What we call produce a high five rate. So it improved the 99 until 90 more than 99% of accuracy. And this method actually is still under development, under developing. So still needs improving. That's why it's not widely used in the market yet. Another third chain sequencing is by this Oxford nanopore. We call it nanopore technology. There is a very small this device. We call it mean ion flow cell. Okay, which have a lot of nanopoles. Very small nanopoles. Okay, and then to sequence the DNA or even the RNA. What they do, they use this motor protein above the nanopore to unwound the double-stranded DNA. When the process of unwinding, the single-stranded DNA, one of it will go through into the nanopore, okay? And the changes of the constant ionic current will be detected and changed to the diagram, and then this graph will be used for the software to this trend, this pattern is a T and then G and then like A like that. Okay. All right. So actually, this is a very good method. Okay, it even promising to show that 400 bases sequencing per second. If really a success method, it will be very helpful to cut down the turnaround time. But this method is also still need a lot of improvement. That's why still not so popular in the market yet. I hope so. It will be a very success sequencing method in the future. I add in actually also you see you no need to add any ENTP other things for it. You just detect the ionic current changes. It also cut down the cost. I can say. The second gen versus third gen sequencing. Definitely second gen is good in accuracy and then the relatively cost effective by now and then the short sequence rate is preferable. For third gen in the future will be a long sequence uh, available and then the high repetitive sequence element. Okay. So this new next generation sequencing panel also can be offered in a hospital setting. One of it example is for MPN or myeloproliferative disorder also. Okay, if you see you observe you can detect multiple mutation site again during the NGS in one run. Definitely, it will request a longer TAT. It's 3 to 4 weeks. This is one of the disadvantages lah. Another example is for acute myeloid leukemia panel. Okay, for using this next generation sequencing. Okay, let's come to case study. Okay, so you expect the same situation previously. Mark leukocytosis, anemia, thrombocytopenia, detected 8% blood with a splenomegaly. Patient have a joint pain, have a low-grade fever. Even though so doctor perform a bone marrow biopsy, flow cytometry, cytogenetic, and even though the PCR. Okay. So the findings for flow cytometry, okay, CD markers 45, 34, 26, okay. So and then the FISH method, okay, also show the fusion gene positive for this PCR ABL. Then the qPCR detected also for PCR ABL. So the final diagnosis can run away is a CML, okay, which this CML PCR ABL fusion gene is coded for this oncogenic thyroxine kinase, which promotes the cell proliferation and decrease the apoptosis of the abnormal cell. So this abnormal cell is difficult to kill and also keep multiplying. So for treatment, usually doctor will the goals will be try to stay in a chronic phase. Okay, if you let it untreated, within 2 to 5 years, usually the patient will become having the accelerated phase and or blast phase, which having the shorter survival rate, the worst prognosis as well. Usually, doctor will issue the first line tyrosine kinase inhibitor, which is imatinib by 3 weeks. So how it works is because this CML enzyme needs ATP binding to activate the further cancer protein. Okay, involved is in cell proliferation and decreasing the apoptosis. But this drug will bind to this binding site and to prevent the ATP binding to it. So there's no downstream process lah. So it improves back the apoptosis level, and there's no triggering of a cell proliferation lah. For treatment, okay, use it for monitoring of a patient. Doctor, again, using this molecular response, okay, which have to be done in every 3 months, okay, using PCR to follow up for this patient, using the international skills to know how well is it the treatment, okay. Maybe halfway the patient is not responding to the treatment, so we have to pick up. So this IS standard is using the standard baseline for PCR LBL at 100%. So a log of reduction indicates the PCR LBL decreased by a certain amount from the standard baseline. Okay, let's talk about log reduction. Okay, if 1 log reduction means actually is a 10% of cells left with PCR LBL gene only. Okay, same to this table. Okay, 2 log reduction is 1%. So 3 log reduction is 0.1%. Okay, so by this table we can know if a log reduction 1 and 2 is usually acceptable for early response, or actually is not ideal for the further period after 3 months especially. So and then log reduction of 3 and 4 is acceptable as a good major molecular response. And 4.5 and 5 is very, very good. We call it deep response and undetectable as well. So by NCCN guidelines, so if we follow this table, we can know how we can plan for the patient. Okay, so you see, if within 3 months is still more than 10% of PCR ABL, actually is not so good, but still can be monitored. But if a rate soon after 3 months is still the same level, actually have to take action already by the doctor. And if it started with a major molecular response, then certainly not respond well, also will trigger doctor to take action. Okay, so the good thing is must be monitor the patient with a good molecular response. Okay, all over the trend. Okay, so for patient management, if patient is a good response, so still will monitor by usually 3 monthly with a PCR, because you need to get the IS skill. The IS skill is useful for monitoring this patient treatment well or not. And then doctor may consider of reduce the tyrosine kinase inhibitor, the drug dosage. Okay, even though the best result is can discontinue the drug. Okay, with criteria lah. So the criteria will be like patient remain remission, remain a deep molecular response for at least 2 years, has no history of resistant drugs, okay, and no advancement phase, means always at the chronic phase. And then the PCR every 1 to 2 months, okay, a patient willing to accept more frequent PCR. And then certain transcript type, means just now I talked about if a P210, okay, is less chance to have a resistance. If a P190, definitely more headache. And also doctor may consider about the lower initial Soker score, which take into consideration of the age, spleen size, platelet count, and then the percentage of blood in your blood. Okay, this actually very easy. It's using the calculator, online calculator. So for patient management, which is not poor response, okay, doctor will take a look on the history of a cardiac, liver, vascular, any issue to this. So then to decide whether want to change to other TKI, other drug. There's a second generation like dasatinib, nilotinib, and the third generation, especially ponatinib. Okay, this new generation of drugs definitely have a proof that is really perform better than the imatinib, the first generation, but uh, it have a more side effect. So that's why doctor want to check with the patient history first. And then if patient totally not respond to the treatment, may consider the stem cell transplant. No? So if let's say patient keep not responding to the treatment, doctor may consider to do the kinase domain mutation check with again the PCR again. So you see by this mutation detection, we can know the special mutation site of each the DNA sequence specifically. So such as a T315I is known case to most type of the drug resistant. And if let's say we find out F317L mutation only resistant to this dasatinib, so actually doctor can consider just issue another TKI lah. So finally we come to the summary lah. So molecular techniques is a trend. So actually it's playing more and more important roles, especially in hematology, to help in an accurate diagnosis, put in a really decision to make the treatment plan, and even though very important for the follow-up for patient. So advancement of molecular techniques, it really well improve the prognosis or survival rate of the patient, especially cancer related. And this is a very sensitive test, but usually more informative if you use it together with the basic like hematology, chemistry, or other area result. Okay, definitely cannot be one man show lah. And future challenges will be the processing time and cost reduction, and maybe the third and fourth generation NGS more mature, and more mutation markers will be come into place. This is the reference I use it for this slide. And thank you. Yeah.
Okay, thank you very much for Lee for the incredible presentations. Okay, so maybe we can move to the questions.
Yeah.
There are some questions in the chat box. I will read the question. So it speaks in Indonesia, so I will translate into English.
Thank you very much.
Okay. Okay, for the first question is from Chris Mayanti. When do PCR used for leukemia diagnostic? And it's not enough only peripheral blood smear?
Peripheral blood
Is it
smear.
not enough only peripheral blood smear, or we have to do always do the PCR or molecular technique for leukemia diagnostic, and when do we use that?
Okay, you see, in a hospital setting, the doctor usually will gather as much information as they can, because leukemia is a very serious disease, right? So usually they will do not only by molecular techniques. That's why I can say cannot be one man show. Still will need the CBC mostly, okay, and like especially like sometimes flow cytometry, okay, because these things is less costly if you compare to molecular techniques. Okay, and also you can find extra information. Maybe it's patient as pancytopenia, just like I say, is it a marked leukocytosis? This one will give you the clues of a patient which to confirm your diagnosis. So that's why doctor will order these few tests to confirm. So, but molecular techniques is tend to give you more details in a DNA level and more specific to confirm the doctor diagnosis, especially into treatment plan, it will be more firm.
Okay, so the uh, sorry, the diagnostic is combined, right? We have combined many diagnostics to decide what treatment is the best for the patient, right? Yeah, that.
Mm,
So we still need the peripheral blood smear, but if we
mm,
want to specific diagnostic, we have to move to the molecular techniques, maybe, right?
yes, true. Mm,
Oke, untuk Krismayanti, jadi cukup jelas ya. Sebenarnya molekuler teknik itu memang digunakan untuk agar kita mengetahui lebih detail spesifik mutasinya itu di mana gitu ya, sehingga nanti treatmentnya itu bisa tepat. Jadi mungkin tadi yang Anda ketahui di slide tadi kan ada beberapa mutasi yang berbeda, kemudian jenis treatmentnya juga berbeda. Begitu ya, Krismayanti, sudah cukup jelas.
Terima kasih.
thank you.
Okay, and then the second question is from Budestri. Budestri. How
Oh,
to do the control quality in PCR methods?
okay. Quality control, usually they will base on their vendor recommendation most of the time. So it seems like almost like a basic hemato and chemistry, they will have their own control material. Okay, even though EQAP also available. Okay, to confirm that especially accuracy is very important, right? So such as very famous in more popular in Singapore hospital that using the CAP, CAP accredited. So they use this EQAP sample to confirm their to ensure their quality also lah. Yeah. I hope so. This answer your question. Yeah.
It's quite clear, Budestri.
Yeah, thank you. Thank you so much.
Thank you.
Okay, so there's okay. Some students like answer your previous slides about peripheral blood smear, I think. So students like answer that is myelogenous acute myelogenous leukemia. Is it correct?
Actually, yes,
Mhm.
because of the blood cell present, but that's why this is one of the possibility. Because blood cell, if you see blood cell only, it
Mhm.
can be a different type of leukemia. Let's say even though you can find it's acute or chronic, uh, so even though have a lymphoproliferative lympho series or as a myelo series, so that's why by itself, just now, the cell identification, I just want to show you that it's not enough for you to make judgment in the specific diagnosis. What disease is it? Yeah, because the treatment follow up by this diagnosis will be totally different. Yeah, but good thing this is one of the possible is a CML. But just now what I present to you, just use it as a example or application.
Okay, thank you, Lee. And I think can I ask you, please? Okay.
Yeah,
I
no
think I'm
problem.
curious about karyotyping because in subject that I teach to students is also about karyotyping, and you know the karyotyping in I think the some hospital is not quite established so,
Mm,
because this technique is tricky and it's so difficult to do.
mm.
And so that's I'm just curious that is there any requirement of peripheral blood for karyotyping? Because maybe be difficult to uh, draw the blood from the bone marrow. So we take
Okay.
the peripheral blood. So is
Mm.
there any requirements for peripheral blood that
Okay.
we can use for karyotyping?
Yeah, actually, I also show in the previous slides. Actually, okay, if you have higher detection rate, okay, actually mostly based on your sample quality first. That's why if you want to detect like cancer-like uh, uh, disease, you have to like especially if your only option is a peripheral blood, it's better to have the blood cell more than 5%. Why? Because there's more mutated cells in your sample. So it will increase your success rate. Yeah.
Thank you.
Yeah.
Okay, because our time is almost finished, right? Maybe we can like take a picture together.
Yeah.
Okay, so please, oke, mohon untuk peserta untuk bisa menyalakan kamera. Kita akan foto bersama. Bisa dibantu ya untuk admin untuk foto. Ya, begitu ya Bu Destri ya.
Ya, Pak Indra yang foto ya,
Oke,
Pak.
oke, mohon untuk bisa menyalakan kamera biar kita bisa foto bersama dengan pembicara kita hari ini.
Sebentar. Please introduce Mr. Lee for Pak Sungging as our head from study program MLT. Pak Sungging?
Hi,
Is
Pak
it?
Sungking.
Yes,
Yeah.
good to see.
Yeah, this is
Nice to
Lee.
meet you. Yeah.
Yeah, nice to meet you too, sir, Mr. Hong.
Thank you again for the invitation. Thank you, University, for the invitation. And thanks for all the time spending with me. Okay.
Yes, sir.
So sorry. Also apologize for the just now I forget to unmute myself. Yeah.
Okay.
Okay, we take a picture. Sudah Pak Indra?
Sudah siap semuanya?
We take three
Ada
times.
3
We have
slide.
three
Yeah, we
slide
have
here.
3. Wow.
We can take a picture for 3 times.
Oh, 3 times. Okay.
Okay, first picture. 3, 2, 1. Okay. Wait.
One more.
Second picture.
Okay.
Ready?
The audience have so many, almost 70.
Good response.
3, 2, 1.
Sudah Pak Indra?
Okay. Last picture.
Last picture.
3, 2. Okay, it's done.
Oke, mungkin saya akan sedikit memberikan rangkuman ya untuk kuliah tamu pada hari ini. Jadi kuliah tamu hari ini membicarakan tentang the role of molecular technique in hematology. Jadi ada beberapa teknik di molekuler seperti PCR, qPCR, karyotyping, serta FISH or fluorescence in situ hybridization untuk deteksi penyakit yang berhubungan dengan darah, contohnya adalah kanker darah. Kemudian juga saat ini juga sedang dikembangkan DNA sequencing hingga 3 generasi sequencing yang sudah established. Tentunya tidak di Indonesia ya, sepertinya sudah established di Singapura. Ya, betul ya. Dengan molekuler teknik ini kita dapat mengetahui spesifik mutasi ya pada kanker darah sehingga dokter itu akan bisa secara tepat dan akurat memutuskan ya mendesain treatment apa yang tepat untuk pasien ya dan juga follow up treatment tersebut begitu. Baik, oke, thank you Mr. Lee for your presentations. Mungkin langsung ditutup ya Bu Destri ya. Oke, sekian dari saya sebagai moderator hari ini. Semoga ilmu yang sudah disampaikan oleh Mr. Hong Fadli bermanfaat untuk para peserta kuliah tamu hari ini. Thank you so much for Mr. Lee for giving the presentations, the incredible knowledge for you with your experience. And so I will close this quest lecture today. Good morning. Oh yeah, good morning. Still morning in here. Maybe afternoon in Singapore.
No, still the same. Just 1 hour different. Yeah.
Oke. Terima kasih kepada seluruh peserta. Saya tutup kuliah tamu
Ya.
hari ini. Dan jangan lupa mengisi link presensi yang ada di chat box ya. Baik, saya tutup. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much,
Oh,
Lee.
thank you so much. Terima kasih. Bye-bye.
Bye-bye.
Bye.
Sudah ya.
Oke.
Terima
Kita-
kasih semua audiens. Semoga tambah pinter-pinter, tambah ini semua.
Bingung.
Nanti kita lanjutkan kuliah tamu berikutnya dari Malaysia atau mungkin juga bisa dari Finlandia.
Mantap, Desri.
Mantap, Bu. Oke, sip.
Oke, selamat hari Minggu, selamat bekerja bagi mahasiswa yang sedang melakukan kerja dan juga selamat hari libur yang libur. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi semuanya.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih, Bu.